All right, in this chapter, we're going to be talking about the molecular biology of the gene. This is a really fun chapter, but it is a very big chapter. So please, if there's anything you don't understand at any point, please feel free to shoot me an email, and I'll be happy to talk over anything or recommend some great tutorial videos. But in this chapter, we're going to talk about the genetic material, the replication of DNA, the genetic code of life, and then we'll go into transcription and translation. So starting out talking about genetic material. We've already kind of talked about Mendelian genetics and the patterns of inheritance, but at this time, the structure and type of genetic storage information was unknown. Mendel's work was highly mathematical and statistical, but he didn't have a microscope to view cells and had no way of knowing how the hereditary traits were being passed on. It wasn't until the middle of the 20th century that researchers started to really understand genetic material. Now, when they first began, they knew that genetic material must be able to store information that pertains to the development, structure, and metabolic activities of the cell or organism. They also knew that genetic material must be stable, so that way it can be replicated with high accuracy and transmitted from one generation to the next. Finally, they knew it also had to be able to undergo rare changes or mutation in order to provide genetic variability. It was the bacteriologist Frederick Griffin that first realized that the genetic material has to have a transformative property. While attempting to discover a vaccine for Streptococcus pneumoniae, he noticed that when grown in a petri dish, S. pneumoniae grew in two different strains. One formed smooth, shiny capsules around the colonies, and the other strain had a rougher appearance. He labeled the two strains S for smooth and R for rough. He noticed that I lost my train of thought. He also noticed that when he injected these strains into mice, only the S strain was virulent. The R strain had no effect in mice at all. So he set up the following experiment. He injected mice with the S strain, and predictably they died. When he injected a heat-treated S strain, the mice lived, and it was known at the time that heat treatment killed bacteria, so he knew that if he injected them with the S strain, they died. If he injected the live R strain, the mice did not die. They lived, they had no protective capsule, or the bacteria had no protective capsule and were vulnerable to the mouse's immune system. When he injected the mice with a heat-treated or heat-killed S strain, the mice also did not die. So then he began to get clever, and he injected mice with a mixture of heat-treated S strain and R strain. And interestingly enough, the mouse died, meaning that the non-virulent strain, so this rough R strain, was transformed to the virulent version by the presence of the heat-killed S strain. So he knew that this S strain was effectively killing mice, and it was effectively dead. It was treated with heat. However, somehow the genetic material or the genetic information from this S strain was transformed into this R strain, and eventually it killed the mice. And when he took a sample from the mouse, he found that the mouse was completely infected with the live S strain. So from this, he concluded that traits could be transferred from one strain to another. This process was known as transformation. So then the next big step was to figure out what this transformative component was. The two main contenders for genetic material were proteins and nucleic acids. Proteins were favored by many in the scientific community because they were comprised of 20 different amino acids, which seemed to indicate a greater opportunity for genetic variation than only the 4 nucleic acids found in DNA. But it wasn't until 1944, when a paper published by a group of researchers, that DNA was discovered to be found the transforming substance. And the way they did this is by using proteases. Proteases are enzymes that digest protein. Strains of bacteria that have been chewed up and digested by proteases were still able to transform non-virulent strains. Strains that had been digested by DNase, the enzyme that breaks down DNA, lost their transformative abilities. Thus, it was concluded that DNA from dead cells was being incorporated into the genome of living cells, or that basically DNA must be the transformative material. Because when exposed to proteases, when we chopped up the proteins, we're seeing no genetic transformation. When we chopped up the proteins, we saw genetic transformation. When we chopped up the DNA, we saw no transformation. So therefore, it must be the DNA that is the transformative substance. So now that DNA was identified as transforming material, the next step was to determine what the structure of DNA was and how it contributed to the coding and the encoding and passing on of genetic information. It was known at the time that DNA was comprised of 4 nucleotide bases. 2 of these bases were purines, which were composed of a double ring structure. This is adenine and guanine. 2 of the nucleotide bases were pyrimidines. These were comprised of only a single ring structure. This would be thymine and cytosine. So then more research had to be done to further understand the structure of DNA and how it works for encoding information. Erwin Chargaff used new chemical techniques to analyze the major components of DNA, and he discovered 2 very important features. The first was that the amounts of adenine, thymine, guanine, and cytosine in DNA vary from species to species. However, they are constant among members of the same species. So the relative amount of adenine, thymine, guanine, and cytosine are going to be roughly the same across all humans. However, they are going to vary greatly from species to species. In each species, however, he noted that there are equal amounts of adenine and thymine. So if you're looking at a fish, let's say that, the fish is 25% adenine, the fish DNA is 25% adenine, it's also going to be 25% thymine. He also noticed that maybe in humans, human DNA was 30% adenine and also 30% thymine, only 20% guanine and 20% cytosine. So he noticed that there are always going to be equal amounts of adenine and thymine, and there's always equal amounts of guanine and cytosine. All of this suggested that DNA uses complementary base pairing in order to store genetic information. So here's what DNA sort of looks like on the molecular level. These are going to be our nucleotide bases. We have adenine over here, guanine, thymine, and cytosine. Down here, we have the relative compositions of each nucleotide base found across the species. So in humans are roughly 31% adenine, 31% thymine, roughly 19% guanine, roughly 18% cytosine. However, in fruit flies, the numbers are going to be much more even. 27% adenine, 27% thymine, 22.5% guanine, and 22.5% cytosine. So these, I'm not going to go through all of these, but these ratios vary from species to species. So the next thing we had to do was figure out the actual structure of DNA. This was going to be done through X-ray diffraction. Rosalind Franklin studied the structure of DNA using X-rays. She found that if a concentrated viscous solution of DNA is made, it can be separated into long fibers. And under the right condition, those fibers can produce an X-ray diffraction pattern. She produced several different X-ray diffraction photographs. These provided evidence that DNA had the following features. 1, that DNA was a helix, meaning that it had this sort of wound around spiral structure, and then that some portions of those helix are repeated. A colleague of Franklin's, Wilkins, showed her photograph to James Watson, who understood the significance of DNA structure. The two individuals who received the Nobel Prize for discovering the DNA structure, Watson and Crick, were model makers. They used Rosalind Franklin's X-ray diffraction photographs to build a model of DNA and publish their findings about the structure of DNA. So this right here that you see is the Watson and Crick model. It's a double helix model, very similar to a twisted ladder. It has sugar phosphate backbones that make up the sides. So we have alternating repeating units of sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way up the sands. They also noted that the two strands of DNA are anti-parallel. Basically, that whatever is being read on one side, the exact opposite is on the other side. So it's basically like a negative image of one side. They also determined that information in DNA must be read in a 5' to 3' direction. So DNA is going to be replicated in a 5' to 3' direction. And we're going to talk about that a little bit later on. But just note that here we go. 5' means that you start with a phosphate, move to a sugar. Phosphate, sugar, phosphate, sugar. The 3' end starts with a sugar, then moves to the phosphate. DNA is always going to be read in this direction, and therefore it must always be replicated in this direction. But we'll talk about that a little bit later on when we talk about DNA synthesis. Finally, they determined that complementary base pairing ensures that a purine is always bonded to a pyrimidine. So adenine is always going to be bound to thymine, and cytosine is always going to be bound to guanine. They received a Nobel Prize for their work in 1962.